Welcome to the third video of skeletal muscle contraction. In this one I'll be explaining cross-bridge cycling. So this takes place in the sarcomere. Pictured here we have a relaxed sarcomere on top and a fully contracted one on the bottom. So I'm going to explain how it gets fully contracted. So it all begins over here in the thin filament when calcium, which was released from the terminal cisternae, comes and binds to the troponin. This causes the troponin to change shapes, which moves the troponin-tropomyosin complex. You can see here that the tropomyosin covers the myosin binding sites on the thin filaments when the sarcomere is relaxed. Once the complex is moved, it allows myosin heads, which is indicated in blue here, to connect to the myosin binding site on actin, on the thin filament. Then to move the thin filaments closer to the middle here, as we have in the bottom one, the myosin head will do a power stroke, which just pulls the thin filament closer to the middle. Then the myosin head will release due to ATP binding to it, and then it'll be at rest, and it'll repeat this process to move the thin filaments into the middle. So this whole process from Z disc to Z disc is called the sarcomere. The Z disc is what anchors these thin filaments. The A band includes the entire thick filament and part of the thin filament. The M line is the anchoring for the thick filament in the middle, keeps everything centered. The H zone is the space that only includes thick filament, and then the I band is the space that only has thin filament. So you can see these in a relaxed one. So down here in a contracted sarcomere, the M line remains the same, and so does the A band. The Z disc, while they are the same, will move closer together due to the thin filaments overlapping. And the I band and H zone actually disappear in a fully contracted sarcomere. And that is all for you guys. Thanks for watching.